Okay, so next TCR gene organization and recombination. So the structure of TCL we have already studied the antigen binding site, variable region and constant region. It is almost similar to that of immunoglobulin. Okay, this I am not going to explain again. So the structure of T cell receptor. So the membrane-bound T cell receptor chains are organized into variable and constant domains. So similar to immunoglobulins, the TCR domains are similar to those of the immunoglobulins, and the V region is a hypervariable region. So it is almost the TCRs and immunoglobulins are almost similar. And the TCR germline DNA is organized into multi-gene families corresponding to the alpha, beta, gamma, and delta chains of TCR. Each family contains multiple gene segments that you can see here. So the alpha chain, beta chain, gamma chain. Okay. So these are location of the chromosome 14 is given, chromosome 6 and chromosome 13. So this is in case of my mouse. Okay. So next, this is just the what is the germline DNA given. So next, you have already studied the organization of the immunoglobulin genes, right? Recombination and all. So similar way here also, I am not going to explain in detail this one. Similar way here also, there is not much difference between the TCR gene organization and the immunoglobulin gene organization. So here also the germline alpha chain DNA, and this is the rearranged DNA and the protein product, and this is the alpha. And similarly, the germline beta chain DNA, this is, and the rearranged beta chain DNA, and it will end up with the beta. So variable alpha and the beta. So this is the T cell receptor you will end up with. So the rearrangement and all, it is similar to the immunoglobulin gene rearrangement. Same way it is. So this is explanation given here how it is. And this is the rearranged alpha chain gene and the rearranged beta chain gene, which is given here just for understanding. So next, a generation of diversity of the TCR. So you have studied antibody diversity in immunoglobulins, diversity, generation of antibody diversity in immunoglobulins. So similar way here also, the generation of antibody diversity takes place. It follows all the methods of immunoglobulin, which follows to generate the antibody diversity. Here also same, but only changes the somatic mutation, somatic hypermutation does not happens in the TCR. Somatic mutation is not used to generate the diversity in TCR. Okay, so except this one, all other processes are same. That is combinatorial diversity, junctional diversity, addition of P and N, P nucleotides and N nucleotides. So all these are same. So I'll give you the overall view of this. So the diversity are generally similar to those that generate the antibody diversity. All the somatic mutation does not occur in TCR genes as it does in the immunoglobulin genes. So here I'm giving the comparison of generating diversity in TCR genes and the immunoglobulin genes. However, the P region nucleotide addition occurs in both TCR and the IG genes, not in the TCR. And the combinatorial association of the expressed change generates additional diversity among both TCR and the IG genes. So same mechanism except the hypermutation. All others are same. So I'm not going to explain in detail about those methods. So you have to learn from there only. So only the differences I am telling here. So this is the how the T cell receptor and the immunoglobulin. So comparison is given here. So combinatorial VDJ joining, alternative joining of this segment, same comparison is given here. Junctional flexibility, N region nucleotide and even P region, here it is not shown, P region nucleotide addition. So all this except the hypermutation. So hypermutation does not take place in the TCR gene, but it takes place in the immunoglobulin genes. So this is the T cell receptor complex. TCR are associated with the CD3. So just for understanding here, I am giving this. So TCL receptor is closely associated with the CD3, a complex of polypeptide chains involved in the signal transduction. Okay, TCR is associated with the CD3 molecule. So this is explanation for that. And the T cell expresses the membrane molecules including CD4, CD8 that differentiate the T cells as cytotoxic and helper cells, and other molecules CD2, LFA1, CD28, CD45R. These are all which play accessory role in T cell function or signal transduction. So just the molecules which are associated are given here. And then this is the general structure of CD4 and CD8. So this is the structure of CD4. Only one polypeptide is present. So these are the co-receptors D1, D2, D3, D4. The what is that domains. And CD28 consists of alpha and beta single domain, like immunoglobulin fold. So this is the structure of CD4, and this is the structure of CD8. Just for understanding this. So this is about the TCR organization and recombination. So in that, all the processes are similar to immunoglobulin genes. Only change here is there is no somatic hypermutation in the TCR gene. So next, the last part of this immunology, the salient features of T-cell mediated cell signaling. So only features they've asked. So I've given extra so that you can understand better in the the process. So when the TCL receptor interacts with its cell-bound antigen receptor, co-receptors, and signaling molecules cluster into the cholesterol-rich lipid rafts of the plasma membrane. So the SRC family of tyrosine kinase, LCK. So this LCK is normally found associated with the CD4 and CD8, and the association between LCK and CD4 is particularly very close. So this one, it starts here now. The first one, cell signaling. The engagement of the MHC peptide initiates the processes that lead to assembly of the signaling complex. So this is the class 2 MHC molecule. So it may be CD4 or CD8. This is CD4 or CD8 co-receptor, and this is the peptide which is associated on the class 2 MHC molecule, and this is the TCR. So these are the co-receptors found. CD3 and all, CD3 which is associated with. So the engagement of MHC peptide with the TCR leads to the clustering with CD4 or CD8 co-receptors. So this is the CD4 or CD8. So co-receptor, this associates with this. As these co-receptors bind to invariant regions of the MHC molecules, LCK is a protein tyrosine kinase. This is LCK. LCK is a protein tyrosine kinase. So this expansion are given in the next slide. So what is LCK? LCK, a protein tyrosine kinase associated with a cytoplasmic tile. Cytoplasmic tile, this one, associated with the cytoplasmic tiles of the co-receptors. This is cytoplasmic tiles of the co-receptors is brought close to the cytoplasmic tiles of the TCR. Okay, this one, cytoplasmic tiles of the TCR complex, and phosphorylates the immunoreceptor tyrosine. So this interacts with this and activates the ITAMS, phosphorylates the ITAMS. Okay, so this CD4 8 associated LCK phosphorylates the ITAMS of co-receptors. Okay, so that creates a ZAP70 means zeta associated protein of 70. Okay, this is the so that creates a what is known as docking site for the ZAP70. So ZAP70 binds here and undergo phosphorylation. Okay, so this phosphorylation activates many number of proteins. So ZAP70 phosphorylates the adapter molecules that are associated. Okay, so phosphorylation of a number of membrane associated adapter molecules which act as anchor points for the recruitment of several intracellular signal transduction pathways. So one set of pathways involves the formation of the enzyme phospholipase PLC, which anchors to the adapter molecule, and it is activated by phosphorylation and cleaves a membrane phospholipid to generate the second messengers like IP3 and calcium. So another set activates the small g proteins that I will show here. So this is ZAP70 after phosphorylation, which activates the SLP67 and the LAT. So LAT is associated with the membrane. So phosphorylation of this creates a site for the many of the enzymes like PLC gamma, GEF, GATS, etc. So this further activates the PIP2 and generates the IP3 second messengers and DAG. So this IP3 which further activates the PKC mediated, that is protein kinase mediated pathways, calcium mediated pathways, and the other one GEF, which so this is one set of pathways, and other set of pathways activates the small g protein mediated pathways like RAS pathway and RAG pathway. So all these pathways finally leads to the changes in the gene expression, the functional changes, differentiation and activation. So this is given here. So one set activates the second messengers. Another set activates the small G proteins. So these are the one. So the second messengers, and this is the G protein pathway. So finally, all activates the leads to the changes in the gene expression, functional changes, differentiation, and activation of the T cells. T cell, because this is a T receptor, right? So this is explanation given here. Um, so what I had explained, same thing is only given here. You can go through once. So finally it activates the ZAP LAT. LAT means it is a linker protein of activated T cells. So all the expansions are given in this slide. So what is GRAB, SLP, ITK. So all these abbreviations are expanded here. Okay. So you can go through once what it happens. So finally, the release of calcium, secondary messengers, and this is the G protein activated pathways, and finally leads to the transcription factor upregulation, reorganization of the cytoskeleton, and cytokine secretion. The final, that is the end of the signaling pathway. And also it affects the expression of adhesion molecules such as integrins on the cell surface, chemokines, which are subsequent effects on the cell localization. So this is about your T cell organization and some of the features associated with the T cell signaling pathways. Okay, so TCR and IgG are same, so only except somatic acquired mutation and signaling of TCR, which leads to activation of many of the proteins. Secondary messengers are activated as well as the G proteins mediated pathways are activated, so that increases the T cell maturation, T cell activation and maturation. Okay, so with this I'm finishing your complete molecular immunology.